All right, welcome to the webinar today. We're going to be discussing proliferation analysis with FCS Express version 4. So we're going to take the next half an hour to give you a brief overview of what proliferation analysis is, how it can be accomplished and modeled in FCS Express, and how you can use FCS Express to make the analysis of your proliferation data a little bit more simple, a little bit more easy to use, and get results quickly and easily. So let's first start by talking a little bit about proliferation analysis, and I'm going to bring up a little diagram here of a typical proliferation analysis setup here. And what I want to say is that the introduction of fluorescent cell staining and division tracking dyes has made it possible to monitor the number of cell divisions during proliferation. So this means we can examine the relationships between proliferation and differentiation and look at different subtypes or phenotypes of cells as they proliferate. So software such as FCS Express is going to help us better describe and analyze that data by mathematically modeling it in a way that's simple and easy to use. So what I want to mention here is that the way that proliferation analysis works is that with each cell division, well, first off to start, what you do is you can stain a cell, or you can add a dye to a particular population of cells that's going to label those cells. And with each cell division, the dye is divided roughly equally between the two daughter cells. Therefore, the dye intensity is actually a measurement of the number of divisions that any cell has undergone. So you can see here when we look at this figure, we have an amount of purple dye. In the second generation after this cell divides, it should have half as much dye as the first generation. As the cell divides again into two more daughter cells, in the third generation for 4 cells, they will have half as much as the second, and so on and so forth down the line. So after the staining, the cell population can be analyzed at regular time intervals by flow cytometry, and generally a histogram of the number of cells as a function of the dye fluorescence intensity is generated. So this is a crude mock-up of kind of what one of those histograms should look like, and we'll show you this data in FCS Express as well. But typically, cell proliferation dyes can be used to track up to about 8 rounds of cell division before the fluorescent intensity is reduced to the autofluorescence of the unstayed cells. So eventually that dye volume that's in the cell will eventually dilute itself out, and you won't be able to track that anymore. Now, I also wanted to mention quickly about different fluorescent tracking dyes. Essentially, fluorescent tracking dyes can be categorized into 2 major groups. One of them is general protein labeling dyes, and if anybody has done any proliferation analysis, you've probably heard of CFSE, which is carboxyfluorescein diacetate succinimidyl ester. And essentially what CFSE and other general protein labeling dyes do is that they form random covalent bonds with amino groups on cellular proteins. And this results in a highly fluorescent cell membrane impermeable marker. CFSE is actually very, very bright when it's excited with a 488 laser, and it emits in the green channel at 515 nanometers. Things to consider with CFSE is, again, it can be very bright, which means it can have a lot of spillover into second and third channels like PE, Sci5, and APC. But then again, since it is very bright, you are able to measure the maximum number of cell divisions because you have a longer amount of time before that dye dilutes out. Now, general membrane labeling dyes are another class. These dyes are dyes such as PKH and CellView, and these are typically lipophilic compounds that stably but non-covalently insert into the plasma membrane. So these generally label the plasma membrane just by intercalating into it. And the general membrane labeling dyes vary in fluorescent intensity. They can also be obtained in many colors. So they make general membrane labeling dyes like this are very useful for multicolor experiments. And actually today we're working with Cell Trace Violet, which is a general membrane labeling dye. So that being said, that's a little bit of background about proliferation analysis. We're going to show you how we can actually analyze that data in FCS Express. And again, if there's any questions as we go along, feel free to ask them in the questions box in the GoToMeeting panel. So let's start by taking a look at some proliferation data. I'm going to open up a new layout in FCS Express. And again, today we're going to be focusing on proliferation analysis, so some of the basic things like inserting plots and statistics and creating gates. We're not going to really go over that too much. We're going to jump right into the analysis. So what I'd first like to do is add some data to my data list. I happen to have a proliferation analysis data set here that I want to look at. And in this data set, we're going to be looking at an unstained control, so cells that no dye has been added to, a cell trace violet positive control, and what this is an undivided population of cells that have been dyed or have had the dye cell trace violet added to them. So this is going to give us a starting point of where our cells that have been undivided are going to begin, and the unstained control is going to give us the endpoint for this particular assay. So we're going to set up a few pages here to actually begin analyzing this data. So on our first page here, I'm going to call this our unstained control. I am also going to set up a second page for our positive control and a third page for our sample analysis. And again, this is just one way of doing proliferation analysis in FCS Express. I think you're going to see when we get to the end of this why we've set it up in this way with our unstained, positive, and sample analysis on 3 pages. But we're going to begin by creating a few plots for this data. We're going to insert a color dot plot for the stained control. I'm going to create another plot and look at side scatter versus a dye called Cytox Advanced. So by looking at scatter and looking at this Cytox Advanced dye, what I can do is set up some gates, and I'm going to call this my scatter gate. We'll insert a gate view so we can look at that, and then we're also going to set up our Cytox dye gate here. And what this Cytox dye gate is going to do for us is any of the cells that have not taken up this Cytox dye are going to be considered live cells. And what we can finally do here on this page is insert a histogram, and we can start looking at our proliferation parameter, which is going to be our cell trace violet parameter there. And you can see what this actually looks like. So what I want to do is clean up this histogram. I just want to look at live cells in this analysis. And you notice here when I apply my live cells gate to the histogram that we are only now looking at the cells that are living, that have taken up the dye. So we have a very nice starting generation for our proliferation analysis that's very clearly defined. Now what I want to do with all of this data is I want to copy all these plots and I want to put them on my positive control page. And actually I should rearrange this data since this is actually my negative control. Let's change these plots to my unstained control here. Right? And again, here's our positive control page, and we're going to change this back to our positive control that we had just set up. So what I did here was just set up 2 pages. One page is going to have our unstained controls where we can actually set up our Cytox gate a little bit better. And what I'd like to also do on these pages is create some markers on our unstained and stained populations. And you'll see why I'm going to do this later, but we want to get a better idea of the median fluorescence for the unstained control, and we want to get an idea of the median fluorescence for the stained control. And we've done that by creating these markers here. So we have a very basic analysis of our stained and unstained controls here, and what we want to do then is create one last page, and we're going to come over here to our sample analysis page. We're going to change the data on these plots to our stimulated control, And then we are going to insert a proliferation analysis plot for our actual stimulated control here. So when you're ready to insert a proliferation plot in FCS Express, it's very simple and very easy to do. You can come up here to the Insert tab and Proliferation. We draw the proliferation plot on the page just like you would an object in PowerPoint. And you can see that we're actually trying to model this data as soon as we put that in. So one thing you'll notice is we're on the forward scatter parameter, which probably won't be of much use. So let's change that to our cell trace violet parameter. And you can see here that we're starting to model this data. And again, what we have to do is apply our live cells gate to this proliferation analysis plot. And then we're starting to look at what most people associate with a proliferation experiment. Now, a nice thing about working in FCS Express, you can see here there's a large amount of dynamic range that's not really used on the low end. If I want to right-click and magnify the resolution of this plot, I can actually just zoom into this area that I'm interested in, and we're going to get a better view of what that data looks like. Now, I'm also going to do some housekeeping on these other plots on my page. Essentially what I want to do here is when I later on run a batch process, I don't want to change this data. So I'm just going to format that real quickly here before we go on. And again, what we've done on the sample analysis page is we've set up our live cells gate, we've applied that to our proliferation plot, and now we're ready to start working with proliferation analysis. So let's talk a little bit about what is being modeled in this data and what the model is actually showing here. You may notice in this plot we have not just the histogram black outline here, we also have a red line. This is showing us, the red line is showing us the shape of the fit that FCS Express is determining. Each of the green histograms or green peaks underneath these histogram peaks are actually the generations of division that FCS Express is determining. And you'll also notice that there's some blue area in the background here, and that's actually calculating background noise in the assay. So those are the three things that we're displaying on this plot at the moment. And I want to also give a brief description of how the fitting algorithm for this data works before we go on. So what's going to happen is the fitting process for FCS Express, or with this proliferation analysis, starts by determining where the population of cells that have not divided is. So that's going to be our population that's located about here. This is our undivided peak, and if we switch back to our positive control, we can see that that's essentially where our undivided peak is. And once it finds this peak, we can try and find it automatically, and if the software will not find that automatically, you can also specify that manually. In some cases, you do have to specify it manually if there aren't enough cells in the undivided population. That's one reason, or if you have a very good control where you have defined where the positive population is, you can set that manually. We're going to show you how to do that in a few minutes. So once that undivided population over here is found, the model attempts to find populations that have decreased in fluorescence intensity by 50% from each other. And since not all experiments work according to theory, and not all biological data actually works exactly according to theory, the model starts looking at the 50% point, and it can move left or right to find the true peak. So as we divide the cells and we go down the line here, we're trying to find the peaks. We're moving a little bit left and right to find out where that peak is, and we can control finding these peaks in some of the options for the fit. And what I'm going to show you now is how to actually adjust some of the fit parameters in the proliferation analysis module to actually make the proliferation analysis fit your data more accordingly. And again, this is because not all biological samples follow the mathematical model exactly. Anybody who's done any kind of biology probably knows that, and you do have to make some minor adjustments sometimes to make the data fit a little bit better. So one thing I do want to do with this data, and I mentioned that we can manually set the starting generation and the background generation, we do have these unstained controls and these positive controls that clearly delineate where those populations should be. So I'm going to right-click on this plot and choose Format. And in our formatting window in FCS Express, when you're working with a proliferation plot, you have a section here for proliferation fit parameters. And these parameters give us a whole lot of options so that we can come in and change how this model is fitting our data. And since we have a known starting generation and a known background noise generation, we can actually use that data to fit our model better. So I'm going to choose here to use a fixed starting generation. I'm going to check this box. I'm going to delete the number that's there. And what I'm going to do is use a token. This little T here stands for token, and that's a live updating statistic in FCS Express. And what this is going to allow me to do is choose the histogram that contains my positive control here. And I can say that for my starting generation, I want to use the marker and the median of that marker as my starting generation. So FCS Express is going to look to that plot and that marker and that median that we've defined in a positive control to use as our starting generation. And now we can do the same thing for the background noise. Since we know we have a negative control that we've defined in this histogram, what we can do is, again, choose that marker and the median for that negative control. And I'm going to click OK and show you how this all works in real time. Now, you may have noticed that this model jumped around just a little bit, and what I'm going to do is do a two-page view, and we are going to view our positive control on the right, and we're going to view our proliferation analysis on the left. So what will happen now is, since this proliferation analysis is linked directly to our positive control, as we move this marker, I want you to notice on the left how the proliferation analysis plot is going to be updating in real time as this marker is moving. So you can see if we move this marker and attempt to find a median value that doesn't quite make sense for this assay, the proliferation analysis algorithm or the fitting becomes all kind of crazy on us, right? But if we select the marker or select the marker around the positive control that we're interested in, we actually have a very nice proliferation modeling, and it's based directly on the data from our positive control. And again, for our unstained control, if we were to adjust this marker here now, you can see that this is going to change how our background noise is going to be calculated for our assay. And if we center this right on the peak, again, we're going to have a very nice fit of that data. So that's just one way that you can use tokens and you can use the fit formatting to actually better fit your data based on known positive and negative controls, which is a very important thing. You're not just kind of eyeballing where that starting generation in the background is, you're actually basing that on known controls. So I want to show you about some more of the fit parameters that fall within the proliferation analysis. And we'll just kind of go down this list and explain what these are. So in our modeling, we do need some information up front, right? We do want to have an idea of how many peaks we're going to be looking for, or how many divisions this cell population has gone through before we model that data. So in this case, the number of peaks is 10. That means we've probably let this assay run for about 8 to 11 days, depending on how fast your cells divide. You're going to be looking at around 1 generation per day. But if I were to adjust this value to something like 8 and preview it, you can see how this is going to affect our model. And we're actually saying anything past 8 markers is going to be considered in the background. So we'll set this back to 10. Now, another parameter in here is our used fixed peaks ratio. And the peak ratio is determined by the software, and theoretically a value of 0.5 describes peaks that are half as bright as the last peak. So FCS Express has gone in here and tried to make an educated guess based on the data what this peak value or this peak ratio is. And again, we're at 0.51 here, so we're a little bit away from the 0.5 of the biological model, but it's not something crazy like 0.6 or 0.3 that would indicate there may be some problems with the analysis. And at any time, if you need to change this value, if you want to set your own peak value, maybe we want to look at 0.49 and preview those results, we can see that that's actually going to be changing the fit for us here as we do that in real time. So again, very important to consider. If your peak ratios look like they're a little bit off for whatever reason, you can always come in here and change that a little bit. So another thing that happens is that in order for the model to find the peak, it's going to make certain assumptions about the width of each peak compared to the undivided control. And that means we're going to take into account the CVs, or the spread of each of these peaks. And that's why we have some options here where we say that we can use the same CV value for all peaks. So this is a default, and we're making the assumption that the CV for all the peaks should be very similar. If we uncheck this box, you notice that it's actually going to give us a kind of crazy calculation because we have quite a large CV for portions of our data. But if we leave that box checked, again, you'll see here that we're making a better assumption with this analysis. And if you really wanted to use your own CV value, if you wanted to override what the software is trying to calculate, if we say use something like 5, you say that it's you can see that the software is actually going to try and divide these into very thin peaks based on whatever the CV value you put in there is. And again, you probably want to be careful using that because you want your analysis based on the data and the CVs that are presented by the data. But again, there's always ways to come in here and try and make this data fit better to a biological model that doesn't always perform according to the theory. And the last kind of option that we have here is to use the same peak ratio for all peaks. And again, what this is going to be doing is we're not going to be making any sort of dynamic calculation that, between peaks 2 and 3, the value is 0.51, and between peaks 1 and 2, the value is 0.5. It will use the same value for calculating all the peaks. And when we do that, again, you can see how this is going to change how our data is actually being modeled at that point. So it is something you can include there. And what I always like to mention about using all of these buttons for changing the fit of your data is that the further you get away from the mathematical model or the theory of working with this proliferation data, that one generation is half as bright as the previous, is the further you're getting away biologically. Meaning that if you have to come in here and change everything about all of these parameters rather than letting the theoretical model take place, there may be problems with the assay itself. So again, the further you get away from the theoretical model, the further away your assay may be from yielding a sensible result. So always keep that in mind. Now, one other point about working with proliferation data in FCS Express I'd like to mention is that this data that's being represented in the proliferation plot in the histogram, it's all based on the actual histogram itself and the shape of that histogram. So it's based on what's being represented in the black lines, right? And since the algorithm uses the actual histogram contour to model the fit, this histogram data should not be smooth. And I want to show you what happens if we come into our overlays and we say we want to look at maybe a prettier plot, so we would adjust our smoothing maybe up to 5 or 6. You can see how everything in this model changes, right? So there are very few reasons that you should be trying to smooth this data. The only reason you would maybe want to smooth that data is if you really don't have a lot of events and you need to clean it up just a little bit. You could maybe use the smoothing set on 1 or 2. But for most cases, you should try to allow FCS Express or the proliferation module to do its calculations based on an unsmooth model. So I'm going to cancel out these changes, and that's going to get us back to our original proliferation plot. And I'm going to show you a few things about statistics in the software. Once you have modeled this data, it's actually very, very simple to get statistics from it. All we have to do is right-click on the plot, choose Statistics, and Proliferation Fit Statistics. And we can also right-click on this plot and choose Proliferation Population Statistics. So what this is going to do for us, many of the common things that you're looking for in proliferation analysis, such as division index, proliferation index, the total percent of divided cells, a whole range of statistics is represented here in this proliferation fit statistics. And then in our generation statistics, we are also getting the peak channel of the fluorescence for each of the generations. We are getting the CVs, we're getting the peak ratios, and what's very important for a lot of people is that we're getting the number of cells per generation, and we're also getting the number of original cells that contributed to the generation that you're now on. So we can see here that in generation 5 there's 1,804 cells, and these came from an original population of about 56 cells. So this is something you have to pay for in other pieces of software. Again, the proliferation analysis module in FCS Express comes free with every copy of version 4. So all of these statistics and all of this functionality is available to you if you're using the demo or if you own the software. Now, if you remember in this particular analysis, we didn't just have these controls. We did have some samples to run through. What I want to mention is that any time that you want to start working with this analysis in somewhat of a batch mode in FCS Express, it's very quick and very easy to do this. What I can do here is let's change these data files to our original here. And just like setting up any other batch process in FCS Express, and let me come in here and change some of my options here so we can go ahead with this batch process. And let's see if I can format these overlays. Right. So we can just use the next and previous button to iterate through all of our controls and our actual samples here, and you'll notice that all of these are being updated in real time as we move between these samples. And in our batch process, we're never going to be doing, I hope, one experiment or one sample at a time. We're actually going to be trying to replicate these analysis many, many times. We can come up to our batch processing tab here, and we can do a few things. So let's actually export this analysis, and let's do our third page here to PowerPoint, and we're going to save this to a file called Proliferation on my desktop. And what this is going to do, it's going to save this information or save this layout on this page for our samples to a PowerPoint presentation that you can present to your colleagues. Another thing people are going to want to be doing with proliferation analysis is probably exporting that data to Excel. That way, when you want to go back over this data at a later point, we can actually go back and summarize everything we want to know about this data in Excel very quickly. So if I want to export to Excel, I can export a few statistics just by dragging and dropping these into my Excel spreadsheet. Let's put in the file name. We'll call this the sample ID. And let's grab a few things like the division index, the proliferation index, percent divided. We can control-click on these and drag them right into our Excel table. And maybe we want to also get the peak ratio and number of cells for the undivided generations. We can bring that in there. So we have our batch process all set up, and when we're ready to analyze all of our samples, we just click the Run button. And you notice that we're going to be iterating through the 4 different sample files that we have. We have 3 controls and a sample. And what's going to happen is that PowerPoint and Excel are going to open up, and this is going to happen as soon as it's done here. And they're going to show us our analysis. So here comes PowerPoint. Here's our unstained control. You can see that this is our simulated control, our actual sample, and our unstained control. So we can easily get to PowerPoint with all these results. These are super high resolution and can be used in any kind of publication. And just as easily, we also exported these results to Excel, where we have a summary table of all the statistics we chose to export. So again, very quick and easy to not only analyze this data and model this data, but it's very quick and easy to export this data in a way that you can digest and a way that you can communicate with your colleagues and publish. So we worked on a lot of this very quickly. I know it's a lot to take in just a half an hour, but I wanted to summarize with a few things. We have many additional resources for working with proliferation data. We have tutorials that cover the basics, that cover formatting, that cover batch processing. You can find these on the web. You can also find them in our tutorials here. If we come into our proliferation analysis tutorials, what you're going to see here is that we can show you how you insert these proliferation plots. We have information on fitting proliferating cells, formatting these proliferation histograms so they look really nice for publications, and again coming into the batch processing. Now, another really important aspect for proliferation modeling is understanding what goes into the module so you know what you're getting out of it when you publish that data. So in our manual, we describe the proliferation module in detail. We explain the fit parameters and all the mathematics used to calculate the statistics. In our manual, you'll find in the proliferation statistics section a breakdown of all of the statistics, the equations used for those statistics, and the description telling us what these results actually mean. So this, again, is very useful when you're publishing your data. You can go back and reference the actual mathematical formulas and have some real-world understanding of what's going on mathematically in these formulas and what's going into the algorithms to fit the data. So finally, if you go through those things and you still have some problems working with the proliferation or you have more questions, you can send any questions you have to support@denovosoftware.com. And you can also get a free trial of FCS Express, including the proliferation module, again, which comes free with FCS Express version 4, and it also is available in the demo version. You can get this by coming up to our Download tab, going to the FCS Express demo, downloading the demo, and you can try all of your own data for free for 30 days to see how the proliferation module is working for you. And at that point, or at this point in the webinar, I'd also like to open up the floor for questions. I know we've covered a lot today. So if you could just find the questions and answers box in the GoToWebinar panel or in the chat box. You can ask any of the questions that you have here. And if any questions come up after the webinar, if anything comes up that you think of that you were not able to ask, you can always send those questions to support@denovosoftware.com, and we'll get back to you within 24 hours with an answer. So are there any questions about anything we've seen today, working with proliferation plots, techniques that go into generating proliferation data, formatting these plots? If so, you can just enter them right into the questions box in the GoToMeeting panel. Great, so I think that's going to finish up the webinar today. We'll stay on the line for a few minutes to answer any questions that may come in. But thanks again for attending the webinar today. We hope you took home a lot of information about how to work with these proliferation data in FCS Express. And again, if you have any questions after the fact, please email them to support@denovosoftware.com.